title, its master project's title is neural net derived gene targets for the nervous system and nervous system derived hierarchies for the neural net. I think after the talk, we're going to have the general audience to ask questions, and the committee members will have meet further with Samuel to see if we have more questions. Yeah, without further ado, please start.
Thanks, Yanning. I appreciate the introduction. So one note, just because we're on Zoom, I hope this little bar is not going to be here the whole time, but there's a little, like, I don't know, gold square in the upper left, or upper right, I'm sorry, that everybody can put their little video bubble thing in. But yeah, without further ado, so I'm Sam, as Yanning mentioned, and I'm happy to be sharing with you the work I've done for my statistics masters. So again, we're talking about neural net derived gene targets for the nervous system and nervous system derived hierarchies for those neural nets. So our story, I think, kind of fittingly, let me just get rid of this. Our story kind of fittingly starts with Gregor Mendel, because frankly, we're both talking about genetics today in a way, and he is best known today as the father of genetics, but also in his time, he actually wasn't really recognized for the kind of landmark work he had done. Instead, he was really famous actually as kind of like this master botanist and also statistician. So for the better part of the 19th century, when people thought about his work, that is what they thought. And in a way, we're really not going to be talking about genetics per se today, but instead methodologies and statistics. So I think it is really fitting that we start with kind of recognizing the particular giant whose shoulders we're standing on, and really who enabled this century of amazing discovery, things like Watson and Crick's discovery of the structure of the genome and all of these wonderful techniques that ultimately ended the 20th century off with the Human Genome Project, and very shortly thereafter, starting the 21st century, the Mouse Genome Project. So all of this landmark work kind of led in 2013 to what Nature Methods highlighted as the method of the year, which was single-cell RNA sequencing. And that in turn led to what Nature Methods again recognized as the landmark method of our current decade, which is spatial transcriptomics. And this is really the core application we are focused on for today. So just taking a step back to make sure everybody's on the same page. So what is single-cell RNA-seq and what are spatial transcriptomics? Well, if you look in the upper right-hand corner of this cool microfluidic GIF, you'll notice a little tiny dot every once in a while running by. That's a single cell. So this technique is really what the name says. We are taking individual cells, we are isolating them in these bubbles with reagents that allow us to do sequencing. So we can actually read off the transcriptome, that is all of the genes that are expressed in a given cell and how many of those genes are expressed. Now, a big problem with this technique, at least from my perspective, is that we have these single cells. We are isolating them from the tissue. And I'm obviously not the only one to think this. A whole field that is now called spatial transcriptomics has emerged, which is in essence letting us take intact tissue, as I'm showing you here with some of the tissue we're actually working with in the lab. And we can do old school things like stain it. Here I'm showing you in green a stain for the intracellular space, or red, the vasculature, or in magenta, this is a particular cell type that my lab cares about. But in addition to doing this kind of old school antibody type staining, we can also identify genes. And so I'm labeling here the transcripts, that is the mRNA that is coding for that magenta protein. And you can see in the spaces where there's not really that magenta color, you don't have those transcripts. And this technique is incredibly powerful because I'm not just able to label that one gene, I can label hundreds of them. In fact, 300 in this experiment that we're running. And I'm just showing you here just a kind of a quick vignette of what that looks like to get a sense of physically what our data is. And of course, we can do all kinds of fun statistics like density plotting. But importantly, what we can do is take advantage of other computer vision type techniques to now not only label these individual genes, but we can then identify which cell they come from. and in essence do what the single cell RNA-seq technique did, but without losing that spatial information. So as I mentioned, and as Nature pointed out, this is an incredibly important technique. So 2020 is the year that they said this technique is ready to go. We can start using it for science. 2021, we already have the first region of a mouse brain that has been spatially mapped with it. And just at the beginning of our current year, we had not one, not two, not three, but four separate, very large projects using this technique. And I'll point out again, all published in Nature, which if you're not familiar with, is the best journal in this kind of biologic science. And they have resolved what they claim to be the whole central nervous system of the mouse. But they forgot a part. And that, in part, is the retina. This is what my lab focuses on. And I'm showing you here a cartoon schematic as if you were to cut me kind of through, let's say, like the middle-ish of one of my eyes, and looked in at what the tissue looks like in there. And that piece of tissue is actually a part of your brain, just doesn't live in your skull. And that tissue's main job, maybe unsurprisingly since it's in the eye, is to take photons of light, that is visual information, and convert it into neural information. It does that using a set of different cell classes. So these are things like at the very top of this diagram, photoreceptors, you might remember from like high school bio, even rods and cones. And it passes them through a set of different interneuron classes, horizontal cells, bipolar cells, endocrine cells, to an output class, which are called the retinal ganglion cells, or the RGCs. Now, this kind of diagram is great because you can really see these parallel circuits that are individually processing regions of the visual scene. So that makes a lot of sense. But what you lose when you look at a view like this is the heterogeneity within the classes. So for photoreceptors, for instance, you can probably remember that you have photoreceptors that are tuned to detect specific colors of light. Now, in the mouse, even though these are numerically the most common type of cell, it's actually a very homogeneous set of subtypes. There's only 3 of them, really. Whereas that output layer of cells, we're in a statistics talk, so I'll mention this is about 1% of the total number of cells in the retina. They account for 45 different subtypes. That is, 45 different pieces or kinds of information are being sent from the eye back to the brain. And let me make that a little more concrete. So the eye is not a camera. In a camera, you have a silicon chip that is being filtered by basically pieces of plastic tuned for green, red, blue light, what have you. And that silicon chip then detects the light that passes through those filters. So the information it is storing is a red version of the world, a green version of the world, a blue version of the world. Your eye is not doing that. I just mentioned all of these interesting cells, and only the photoreceptors are in some way akin to what's happening in the camera. Just this first layer right here in what I'm labeling the ONL, the outer nuclear layer. All of these other cells are doing computation. And that means the information going back to the brain is much more complicated than just different colored versions of the scene. How complicated? Well, the Sains and Meister lab have a really important study where they basically looked at the most common ganglion cell type, that is the output cell from the mouse retina. And they asked, if I play a video of normal naturalistic behavior to it, basically just a cat running around with a camera strapped to its head, what are the cells going to do? And the thinking was, of course, that the most common output from the retina should be active all the time in a very reasonable, realistic view of the world. That is, of course, not what happens. These most common tunnel types are those W3s highlighted in red here. And what you should appreciate is they don't fire it all to this video of a cat running around, just looking at stuff. So after quite a bit of work, these authors actually discovered that what these cells do respond to are dots of a very specific small size moving at a very specific velocity. And this is the equivalent of basically what a bird would look like to a mouse. So taking a step back, the output from the most common information channel in the mouse eye is not a color, but it's in fact, is there a bird or is there not a bird? And when we look across the set of all ganglion cells, we can appreciate, without going into the detail here, that not only do these things look differently, their morphology is different, but their physiology is different. And you're seeing here basically different responses to the same visual stimulus to all these different cell subtypes. And it is a very diverse array. So there's a lot of different information coming out of the eye. So that's already different from a camera. But this camera analogy is also misleading because of the spatial information. That is, instead of looking at a cross section through the retina, let's think about looking head on at each other. So in a camera, you have a uniform grid of these red, green, blue filters. And therefore, what you're really seeing is a one-to-one representation of the visual scene onto this array into the information space. Now, our retina, we are primates, has what is called a fovea. This is a high density region, lots of cells clustered together. And that's in contrast to the periphery, where the cells are much more spaced out. And this results in a distortion of the information that can actually be acquired anytime the eye looks at the visual scene. And thus, the information that's sent to the brain is biased based on this spatial arrangement of cells. And this is true for all species. I mentioned that we are primates. If you look across the animal kingdom and you ask the question, what do their retinal topographies look like, where or rather Anhelt and colleagues are representing more cells, so a denser packing of them is darker, you can see that the evolutionary pressures on a given animal really dictate the mapping of its cells. In the mouse, for instance, the top of the eye, when we're talking about optics, everything is flipped. So the top of the eye is looking at the bottom of the visual scene, and it is sensitive to effectively green light. So think grass. Whereas the bottom of the eye, so the top of the visual scene, is sensitive to blue light. Think the sky. So already you can see that evolution is shaping the arrangement of these cells. That is, evolution has a hardware optimization on the computation the retina can perform. And this matters. When we look at a mouse behaving, so in particular, a mouse hunting a cricket, if you measure how its eyes move, what you'll find, as Holmgren and colleagues did very recently, is that the position of the cricket is fixed on a very specific part of the cornea, that is the outside surface of the eye. And when you do those optics transformations, you'll find that that spot corresponds exactly with a spatial cluster of what is called alpha-on sustained ganglion cells. And we know, thanks to work that was actually just published at the end of this year, that those cells are the equivalent cells used for carnivores throughout the mammalian kingdom. And I've been using this term clustering or spatial arrangement, but this isn't a qualitative idea. It's actually very quantitative. So here's a piece of tissue I took that has cells labeled in green. You can probably appreciate those little green dots. And you can certainly appreciate them better when I segment them out. So just relatively simple image processing. But now we have basically the center position of every cell in the retina for this type of retinal ganglion cell class. We can then perform what are called first order spatial statistics and recover something like those topographic maps you were just looking at. And more interestingly, we can perform second order spatial statistics, which allow us to appreciate that when cells are very close together, so close to zero on this distance plot on the x-axis, we have a uniform distribution of these cells. This is well known, in fact. But when you look across the extent of the retina, so we're looking at greater distances between cells, not only do we not have a uniform distribution, what you would find in a camera, we have clusters, which you can appreciate by eye. So here again, I'm showing the classic alpha-on sustained ganglion cells I just mentioned, those cricket hunting cells, and you can appreciate by eye in these guys as well. There's simply more of these cells in this dorsal temporal region of the retina than there are in this nasal ventral. And when I went and I did a survey of all of the different distributions of cells that are known in the mouse and their spatial arrangements, what was found, or what I found, and I'm not the only one, of course, who's appreciated this, that on the one hand, there are lots of very interesting different distributions. There's a heterogeneity. But on the other hand, there are huge gaps in knowledge. We really don't know the arrangement of most of the cells in the retina. And so that's the focus of my PhD work. And how do I even know that all these cells exist? I mean, I showed you that physiology book before, but the real gold standard is actually that single cell RNA-seq technique I mentioned. And with that technique, we have been able to, or rather the Sains lab has been able to cluster all of the different cell classes and find their subclasses, their subtypes. And from that, we know the full atlas of cells in the retina, which brings us to our key question. So how many subtypes can one classify given some number of genes? We know the gold standard is with tens of thousands of genes measured by single cell RNA-seq, one can measure the 125 neurons, or about 130 different cell subtypes in the retina. And before I tell you how we solved this problem, I think it's important to do a little bit of statistics background, just following on that biologic background. This is, after all, a statistics talk. So we have a question, and it leads itself to a nice quantitative solution, right? We should expect something in the form of like X genes can classify Y subtypes. And this proposed solution already gives you a hint about what you should be doing to figure out the answers to that X and Y. And that hint is the word classify. So whenever you hear classify, what you should be thinking is machine learning. And I'm going to quote Josh Starmer of YouTube fame here and say that machine learning is a collection of tools and techniques that transform data into decisions by making classifications. There's that word again. The other word that's really important here that's already come up is data. We have data. In fact, our problem is we have too much data right now. We want to try to minimize this in some way. So what do we need to do? Well, right back to Josh's definition, we need to come up with some kind of transformation. So for that, we will be using, or we will be creating rather, machine learning models. Bam. So what is a machine learning model? Maybe it's worth mentioning what a model is to science in general. Statistics is, after all, just the language of science. And the physicist Byron Jennings has a nice statement, I think, that kind of frames it well, at least what a model should do. And what science is all about is basically the construction of parsimonious, internally consistent models that can reliably predict future observations. So with this kind of framing in mind, let's consider in an abstract the problem of having data and trying to come up with a model. And in particular, we'll talk about making a regression model. So this is the formula that defines that, but really it's just y equals mx plus b, which everybody should remember as the slope of a line from like 7th grade math. So great. So we have a data. We are going to somehow fit it to this model. And that is going to let us do inference, that is, make these predictions that Professor Jennings referenced. But the other thing a model can do is it can let us imagine or predict what an unobserved instance of our data would look like, or even an instance of our data that might not even be realistically possible, but could have some other kind of interesting theoretical importance for us. There's one other component of machine learning models I really want to highlight here, and that is the discrepancy, that is the error. So everybody is probably familiar with the idea that models are useful, but they're not exact representations of the world. And there are lots of reasons for that. But that error, what we would call the residual when we're talking about machine learning, is the discrepancy between the actual observed data and this model, what we would predict for the given instance on, let's say, just the x-axis. So with these fundamental thoughts in mind, this is, after all, talk about neural networks. So what do I mean by a neural network model? Well, the reason I started with regression was because really, you might be wondering, how do you get that green line? That's actually what brought me to this whole statistics master in general. But how do you get this green line? You know, it could be there, sure, that looks nice, but you could do that, you could do that. And with what a neural network is it saying, let's create lots of different models given a set of data. So you can see all the blue dots are in the same place, same data, but you can make different models that are going to make different conclusions based on that data. We can then wrap up these conclusions, these predictions, in what we would call a nonlinearity. And now we can think of each of these different regression models with their nonlinearity wrapper as a node, or what you might call in the parlance of neural networks, a neuron. And these neurons can then make some prediction that you can pass forward to another model. And again, these models can be a set of models. And in the particular kind of neural network architecture we're talking about today, we're talking about a fully connected architecture. So it's not just one model giving its output as an input into the next layer, but it's all of the different nodes in a given layer. And then we can keep playing this game. We can make more nonlinearities and pass forward the predictions from those models to another set of models, which we can, again, keep wrapping in nonlinearities, et cetera, et cetera, et cetera. It's worth pointing out, just to be clear, that not all neural networks are fully connected in this way. And even those which are fully connected don't necessarily have all of these arrows or all of these nodes. And that can happen as a result of techniques like regularization or dropout. Regularization will come up again shortly. But really, let's make it concrete again. So we have data. We have measured individual cells that have some kind of gene expressed in them or not. And we can measure that number of genes, pass that into a neural network model, which will perform in this linear, nonlinear manner a filtering, a prediction, and give us a classification result. In this case, the presumption that this given cell is going to be labeled as a 45 alpha off transient ganglion cell. All right, but so again, talking about where did these models come from, it really has to do with that error, that residual. So we can measure these arrows, right? The discrepancy of the model, that green line, and each of these dots, line them all up. So now we have some number. And we can basically take the average of that discrepancy, and we can call that maybe a cost function. Now, in particular, we're going to call this the mean squared error. And we're going to square these guys really just so we flip every arrow in the same direction so that we're not canceling anything out. But with a cost function, one can perform something called gradient descent, which is a really cool, in a lot of ways, basic calculus trick, where you can make a set of weights. If we remember back that y equals mx plus b, we're talking about that m, or in the parlance of linear algebra, the beta, the weights. And we can tweak that weight in such a way that we can test different models and see how their mean squared error, or whatever your cost function of interest is, relative to the data. And in so doing, we can use gradient descent to iteratively move down a slope, either in one dimension, as I'm showing you here, or in any arbitrary number of dimensions, to find this minima, this point at which the cost function is as small as possible, or reasonably small enough, I should say. And I say reasonably small enough because how do you know when you've actually reached it? The math doesn't necessarily tell you, based on the way we actually perform the gradient descent algorithm, okay, you are at the minima. I can tell you for sure that you are. Instead, what we do is we set a number of training epochs. That is a number of these little red dots you're seeing here, these steps down the gradient. And you can either have some number of steps that you set, or you can have something called early stopping implemented, where at a certain point when your cost function is not improving significantly enough, you decide to terminate your search. Wonderful. Now we have a model. Again, we got those weights from gradient descent, but how do we know it's any good? And that's where signal detection theory comes in. And this is actually, it comes originally from electrical engineering back in the day when we had, faulty wires. But it's something that we've made really nice use of in psychophysics to evaluate how people actually sense things, how much perception we have of something. And the key components, and we use that as well for machine learning. And the key components here are the signal, that is the stimulus or event being detected, the noise, that is the interfering stimuli that you're trying not to detect, and then this idea of a hit, basically correctly identifying a signal, versus a miss, that is failing to detect a real signal. And these are in contrast to a false alarm, when you incorrectly identify noise as a signal, or correct rejection, when you correctly identify that noise is noise. And we can represent these kind of ideas, the signal and noise, with the concept of a probability density function, where on the y-axis, we're basically representing the probability that some event represented on the x-axis happens. So for instance, you can imagine the x-axis being basically frequency or pitch, and maybe the signal you care about is your baby crying. And so as the pitch increases, you have a higher probability of it being your baby, up to a point. Your baby's not going to sound like a kettle. So wonderful. Let's say that is our signal distribution. We're going to draw a criterion. We're going to have a threshold above which we say, this frequency is probably my baby. I better go to the next room. Or below it, maybe the baby has like a cough that day, or the pitch of its voice is lower. Even though it truly is crying out for you, if that frequency is below your criterion, your threshold, you're not going to maybe say that the baby is crying. Now, on the other hand, we have our noise distribution. So basically, think all of the other noises that you could be hearing at a given moment. Cars on the street, the fan in the room, television across the hall, whatever it is. And sometimes you're going to have frequencies which are basically the same as what you would expect from your baby crying. These would be your false alarms. On the other hand, you might correctly identify that your baby does not sound like a car on the street based on the pitch. So with those key components in mind, we have our noise distribution overlaid with our signal distribution in reality. And we have our criterion, that is our threshold for making decisions, somewhere in this space of, let's say, baby crying or whatever it is that we care about on our parameter space. And our goal is basically to differentiate these correct rejections and these hits from the mistakes that we could make. That is the space in which you have both hits as well as false alarms, or correct rejections and misses. And the separation between these peaks is really important. This is what we would call sensitivity. And this is almost identical to the simple test statistic Cohen's D. So not surprising, we actually label this sensitivity with D prime. And it is literally just the space between the center of your probability density functions. And so if you've got two distributions really far apart, you've got a really big D prime. See, on the other hand, our distributions are close together, little tiny D prime. You're much less sensitive to differentiating your baby crying from the other noises around you. And that'll result in these false alarms. So what we're interested in really is the relationship between our hits and our false alarms. When we plot that, this probability of your hit versus the probability of your false alarm, we're going to come up with something that we call an ROC curve, or a receiver operating characteristic curve. And the way you should think about this, if your signal and noise distribution were exactly overlapping, that is, there's no way, you're totally guessing every time if it's your baby crying or it's the car on the road, you're going to wind up with an ROC curve that is a 45-degree line like this. As your distributions, your noise and your signal get further apart, your curve moves further up to the left. That is, you get more sensitivity, or less, again, as you move them closer together. But this is the intuition you should have. And it brings out this confusion matrix ultimately, where you can have basically true positives. So if one were to imagine a doctor diagnosing a very clearly pregnant person as pregnant, that would be an example of a true positive, as opposed to a doctor diagnosing a very clearly not pregnant person as not pregnant. That is a correct rejection. And together, this gives us the simple statistic that most people are familiar with, which is accuracy. But accuracy, as I just pointed out from that ROC curve, doesn't account for the mistakes that a model can make. And when we think about this false positive regime, basically the doctor now telling this very elderly gentleman that he is pregnant, this would be in statistics what we think of as a type 1 error. And the statistic we're going to use to really deal with type 1 errors for machine learning models is what we call precision. Now, on the other hand, you can have what is arguably, in many cases, the worst kind of error one can make, which is a type 2 error. So telling this very clearly pregnant lady that she is not pregnant. And we are going to, in machine learning parlance, look at the statistic called recall, or more formally, the true positive rate. And so I'm just representing here for you the exact formulas, the ratios that we use to calculate these. And I'm going to highlight one final statistic, the one that we're actually going to be focused on for most of the models I'll be showing you. And that is the F1 statistic. This is the harmonic mean between the true positive rate, your recall, and your precision. So just as a final important, or almost final important technical note on this statistics background, I told you about the data that we're using to fit the model. Technically, that's your training data. When we're actually making a machine learning model, we're going to have both training data and test data. And that test data is going to allow us to compute those statistics we were just talking about. And I'm representing it here with these little arrows. This is, of course, not what is actually going into making your F1 rate, but it is very highly related to it. And I just thought it would be more convenient to keep it in this graphical display. All right, so that was a lot of background. I apologize, but we are going to now dive into the problem itself. So now that everybody's on the same page, let's talk about gene selection. So what is the data that we actually have to work with? Well, we have 3 different single-cell RNA sequencing data sets, and I'm just going to run through the RGC data set to give everybody some intuition about what we're working with here. But basically, this data set consists of some number of observations, in the RGC case, about 36,000, And those observations are really vectors of tens of thousands of genes. In the RGC case, 18,222 genes. These are individual genes that have been measured and they have been normalized according to this very relatively common bioinformatic method to create an expression matrix, which really is able to occupy anywhere from non-inclusive 0 to infinity on the reals. In practice, we're really talking more like 0 to 2. And from that, the authors also were able to propose cluster identities. So these are basically going to be our factors that we're trying to predict. And I want to highlight that these are not homogeneous data sets. Even though we have these 45 different clusters, you can appreciate either looking at the cluster view itself, the different like colored blobs, or in this histogram, that the relative proportion of different cells of a given class is wildly different from one class to another. The other final piece of information, at least for the Tron data set that we'll be talking about, is the authors basically looked at their genes and they said, can we give biologists a minimal set of genes? So think 1, 2, or 3 genes tops for a given cell that can identify it. And while they proposed this set of ultimately, or the subset rather, of 61 genes, you can appreciate from this, if you will, identity matrix, or something like an identity matrix, that it's very, very messy. There are probably, at least when we started this project, we thought something like maybe 76 to 98 genes that one would need to do a fairly decent classification of all these different RGC classes. So representing this in kind of little boxy format, we basically got these 2 different pieces of information from a given study. We have that expression matrix, your genes by cells, and we have a vector of labels. We have that again for 3 different studies. And what I did is I simply combined them into a single data frame where we have, again, this total number of genes that one could have measured and all of the cells that were measured. So each row is basically a vector of these genes. And with that, we can create a subset of this data frame where you can have any arbitrary set of n genes for all of these different observations. So that is what we're going to be working with and trying to mine some insights out of. Back to our question at hand. So I mentioned already that Tron and colleagues had proposed a subset of 61 genes. We were interested at the time in trying to classify as many subtypes as we possibly could. And we decided that we wanted to be really strict about this. So we were looking at basically the types of cells that we could classify using a neural network in particular, that had a 90% true positive rate, that's our recall again, and a 90% precision. So what we were focused on at first was this manual regime where one is actually labeling all of the genes by hand. That's where we actually started the project. And RNAscope was the specific technology we're looking at. So you can see here the number of genes that one could measure. And unfortunately, Tron was outside of that. So we had to pick a subset by hand of 10 genes just to start. And we had picked this set of genes in a very clever way. So when we fed it into our neural network, we were actually able to achieve a comparable performance with this very, very strict subtype classification threshold to what the Tron proposal was. And you can see here basically a schematic or a sketch of what these 2 neural networks that we were working with looked like relative to one another. As a technical note, how did I wind up with those specific neural network architectures? Well, basically what I did is a grid search. So I compared lots of different architecture, so multiple numbers of hidden layers, for instance, as well as different training regimes. So we're looking at different cost functions. I mentioned MSE as one example. Part of my work was actually coming up with a custom loss function. As it turned out, a very simple 2-layer neural network with the categorical cross entropy was good enough for our purposes. And through this grid search, we were able to not only find an optimal architecture, but also an optimal set of hyperparameters. So I mentioned regularization, that's going to be that L1 lambda. So I created a bunch of models with different L1 lambdas and a given architecture. And then I trained them for a number of epochs. Remember, that's the steps down the gradient. And I gave them some number of early stopping steps that they could have. And through this grid search, we were able to identify not only the optimal architecture, but the optimal set of hyperparameters for a given architecture. And I'll point out that this involves what is effectively a very simple cross-validation, where every time I created one of these models, so different set of hyperparameters, different set of architecture design choices, I basically rolled the dice 5 different times. That's basically what a random seed is. So what you're looking at here and in the previous graph were basically the mean values, the mean F1 scores for these different dice rolls as we shuffled our training and testing sets. And I'll also point out again that, of course, the F1 scores that we're actually looking at here come from the testing sets, not the training data. So just showing you very briefly, this is basically the performance of our optimal one-layer neural network, one hidden layer neural network, rather. And you can see basically a breakdown of its performance on classifying individual classes. And you can see it's an incredibly high on average F1 score, above 98% for all of the different classes, with in general a pretty good subclass performance. This again was how we were doing our prediction of if we were doing a good job or not. And I'm also showing you here, because we had that cross-validation, the variance between these subclasses over those different cross-validation splits. But we have a problem, as I mentioned, very, very unbalanced data sets. That is, the number of examples of a given factor, a given type of cell, or subtype rather, is not equally represented throughout our data set. The other problem is I'm using this, on the one hand, very simple neural network architecture to do a lot of different tasks, and it's doing them all fairly well, but personally, I didn't really know how to think about how it was doing any given task. So what was the solution? Well, ultimately, I came up with a curriculum training strategy where I basically said, instead of having one neural network that is doing 45 or 130 different tasks with one data set that is training it, that is very unbalanced, what if instead I made 130 different neural networks where each neural network was basically trained to do one specific task by giving it a data set that was 50% signal and 50% noise. I could then take the gradient, remember that step down, that way we were training the model, to rank order every gene and decide how important a given gene was based on its gradient for the classification of the target subtype. And in this way, we were able to look at subsets of genes within the regime of spatial transcriptomics. And these are kind of how, at the time that we were doing this, the different spatial transcriptomic technologies could measure genes. So with this approach in mind, I basically was able to now take this rank ordered list of, call it 1,000 genes, and I could say, okay, for 50 genes, how many subtypes could I measure? For 100 genes, for 200 genes, for 300 genes. And ultimately what we found based on this unbiased gradient search approach, gradient selection approach rather, was a plateau around 225 genes. The technology we're using is the 10x Xenium, so we could actually add in about 75 extra genes that were interesting for biology. How good is this though? We're using very simple statistics again, this true positive rate and precision to say that we're doing a good job. We're actually doing quite well. So this is a projection into t-SNE space and another projection into UMAP space. On both of these, what you're looking at are the same projection of all of our cells in the test data set that are colored based on their subtype. And I just have the plot replicated 4 times so that you can appreciate the different classes are clustering one to another. And the different subclasses, and I'll blow them up here, are also clustering one to another. Basically, what you're looking at is the color of these different blobs, if you will. The more pure the color is, the better the classification performance was. So this was a really nice way of appreciating that not only were these simple statistics, these high true positive and precision thresholds being met, but the actual pattern of the data is being preserved. You can also appreciate that when you look at the gene expression matrices, which is a more common bioinformatic way of looking at this, where you can now look at the 300 genes we selected, the 225 from the plateau and our 75 for biology. And you can appreciate a region that is basically a bunch of a bunch of ganglion cells. Another region that's a bunch of amacrine cells. Here are the bipolar cells. Here we have photoreceptors. Over here we have Mueller glia. So these are nice ways of checking that our statistics are reproducing the biologic patterns that we actually care about. There's one more problem though. So now we need to do inference, but what you might have noticed is that the number of genes from each of these different atlases is actually quite different from one to another. And with the neural networks, I mentioned that we have this input layer that is basically taking in this vector of information. And that input layer is taking in numbers. It is fixed how many of these numbers and which specific numbers it can take in. So what we have to do is we have to somehow get these vectors to all be the same length in order to use a neural network. So I did something called zero imputation. If you imagine, for instance, gene 2 was not measured in data set 1, I'm just going to put a 0 every time that gene 2 is mentioned in regards to data set 1. Likewise, you can imagine maybe gene 3 wasn't measured in data set 2. It gets zeros. Gene 4 actually wasn't measured in data sets 2 or 3. Zeros again. How about gene 5? Well, it wasn't in data sets 1 or 3. So again, zeros. And the problem here is that one can very easily imagine the neural network that I was showing you with all that nice performance was actually just cheating, and it was identifying what data set something came from based on the presence of zeros in these specific gene columns. It doesn't actually know what genes are, of course. So how did we deal with this? Well, the first inspiration came from really the classification game that one would play in the Biologic lab. When one wants to classify a given cell, the first thing they're going to do, as I showed you before, they're going to draw a circle around the cell. They're going to say, okay, here it is. And now we're going to say, in a hierarchical manner, does it express the class marker? Something like RBPMS, which I mentioned at the beginning of the talk. You can see all of the RGCs here, these orange dots, they all express RBPMS. That's why we know that they are RGCs. So great. You can then add on iteratively more and more genes and very quickly create a barcode that tells you a given cell based on the genes it is expressing is a member of a given subtype or subclass. So that's our first bit of inspiration. The second bit, because now we know we want something hierarchical, is the cephalopod nervous system. And I'll be talking about cuttlefish, but they're all organized in a similar manner, where you basically have a central ganglia, which is akin to a brain that can do things like see, it has eyes, and it can direct the organism, the octopus, the cuttlefish, what have you, to maybe do something like go for a fish. But each of those individual tentacles or arms that it has actually has basically its own brain. These are a network of ganglia, which you can see here in this maybe somewhat disturbing demonstration, that without a brain, they work just fine and they can do very complex behaviors all in and of themselves. So cool. I wanted some kind of hierarchical network that could, in some ways, do something like what a cephalopod can do, where each individual network, while related and while able to communicate to some extent with each other, are doing specific behaviors. So we have a working neural network architecture already. I told you about all that grid searching we did to find the architecture that made sense. And this is just another way of looking at it. We basically have our input layer, we have our hidden layer, and we have our predictions. So with this particular zero imputation problem in mind, what we really want to do is we want to say, okay, the input is only the genes that do not have any zeros that are artificially added in. These are going to be clean data that let's say the head of the cephalopod, that central ganglia, gets access to. Whereas the output now is going to be a subset of actions. In this particular case, that central ganglia is going to be classifying just classes. So ganglia cells, bipolar cells, photoreceptors, that kind of thing. And then we can build, using the same exact architecture, a tentacle for each subclass that we're interested in, where if you notice the numbers here, the inputs to any given subnetwork, any given tentacle, are only the clean genes that can identify the subclass of interest for that specific tentacle. And through the magic of PyTorch, we can stitch this all together and get a nice unified classification. So the output vector is going to be our log probabilities of a given input vector of genes with no information as far as the network is concerned about the dirty genes relative to a given subtype. And I did, of course, another grid search here. I won't belabor the point, but it was done in effectively the same way as the original architecture. And here's just those optimal performance metrics again. I'll point out that with this hierarchical structure, we actually are performing even better. You'll notice that we are well above 99% on all class classifications with none of those dirty genes. It's not using the data set knowledge to cheat. And the subclass information is phenomenal with very, very low variance between my cross-validation folds. But how good is it really? Well, I benchmarked it against the best-in-class similar network. This is also a hierarchical network called Celtics, which is designed to classify cell classes as well as cell subclasses. And we blew them out of the water. Again, since this is, and I'll point out that delta rate here is basically the optimal training version of our single cheating network. So this is, of course, a statistics presentation. I couldn't end it without an ANOVA, now could I? So here we can see that when we compare the class and the subclass performance, we do phenomenal. We blow all of these other models out of the water. And when we do post hoc testing, I did a Tuckies for reference, we're able to reject our null hypothesis relative to CuttleNet versus all of these other network architectures very significantly for the Celtics model, and for our fully connected, that delta rate model, also quite significantly. And you'll appreciate that in practice, the difference between our CuttleNet and our fully connected network really doesn't look by eye like it is. So you're not going to be surprised to find out that the Cohen's d is also quite small. Remember, that's the separation of that signal and noise distribution. So practically speaking, what this really means is that we have developed not only this cool neural network-based gene selection strategy, but we've also been able to match the performance of the much more complicated bioinformatic approaches to clustering cells. Of course, we're doing it in a supervised manner. They are doing it in an unsupervised manner. And then we can overcome the zero imputation problem of combining distinct experiments, distinct single-cell RNA-seq data sets. This is something that is of practical importance with CuttleNet, which outperforms the best-in-class model and doesn't lose any performance with the extra complication that is added by this hierarchy relative to just a much simpler model which, again, might be cheating a little bit. So what are we doing next? Well, we have this beautiful data set that I started the talk off with, and I'm showing you here on the far right our first pass at just performing inference with CuttleNet on the genes we've measured. So now we just have to finish analyzing it. And with that, we are pretty much at time. And I really want to thank, in particular, my committee. I feel really fortunate to have this particular committee because these Yaning, Josh, and Aaron were my professors for pretty much all of the stats classes I took. And of course, would not be doing any of this without Alon, who has been just nothing but incredibly supportive and frankly inspirational in this journey of going from being a pure biologist, which is what I really thought of myself as before, to someone who can do these kind of cool computational techniques. I'd also like to thank both the statistics department and the neuroscience graduate program, and of course, my friends in the lab. I also want to really just highlight Yaning again in particular, because I don't know if this email means nearly as much to him as it does to me. I'm sure it doesn't, but this is really what started it all off. Heart of the COVID pandemic, I could not go into the lab. I didn't know what to do with myself. And Yaning really took a gamble on me. He let me come and hang out in his regression course, and one thing led to another, and here we are. Here's Pipoka, my very loyal companion through all of these classes. She actually was on my lap for probably all of Yaning's classes and most of Aaron's as well. Miley, who also, keeps me happy, keeps me entertained, and my wife, who really keeps me sane and makes this whole thing possible. So yeah, it would be, who knows where I'd be otherwise. But I want to thank you guys all for attending, and I'll take any questions.
Thanks, Sam. It was always great to have you in my classes.
Thank you, Andy.
So yeah, any question from the general audience?
Hi there. I have a question.
Yes, please.
This is Mark in California.
Hey, Marky, how you doing?
Hi, Sam. Hey, you mentioned something about something in your eye that's part of your brain, but it's not really in your skull. What were you talking about?
I'm talking about actually this funny little video I've got playing right now. So this is the retina. Just convenient that you asked that question. I swear you're not a plant. So this is when people say like, the eyes are the window to the soul. If you're like me and you think the brain is your soul, then that expression is actually dead on because you have a little piece of your brain that we call it the retina, and that's what you're looking at here. That is in the back of your eyeball, and that's what is doing all of this light detection that ultimately leads to the computation that Alon's lab is interested in.
So you basically have part of a computer in your eyeball. In the back.
100%. Yes, exactly.
All right, that's cool. Thanks, Sam.
Any other questions from the general audience?
Hey, Sam,
Hey, Mike.
I have a question for you. So you started out the talk, you showed all these different subtypes, different morphologies, and different physiology. But you've reduced this down to just 300 or so genes. There's probably many more genes responsible for that kind of variability in your morphology and physiology. So how does this help? How does this kind of reductionist approach help us understand what you're showing here? Or does it?
No, it's a perfect question. So definitely, like the different genes that one is interested in can probably be used to predict in a much more purposeful way, like the physiologic responses, or maybe even some of these structural shapes that emerge. So by ignoring all of those genes, we lose that information, no question. What we're really doing with this this dimensionality reduction is we're saying what subset of genes can be used just to name a cell. And so when we name a cell, then you can go ahead and you can measure the other genes that you care about. So now you can say already, okay, I'm looking at, let's say specifically a W3 RGC. Now we know it's W3 RGC because of the set of 225 genes that we predicted. I mentioned that we added in 75 genes for biologic interest, and that is exactly kind of what you're getting at. So those extra genes that we're measuring, or maybe those extra genes in some of the 225, they're not mutually exclusive set, can be used to predict some of these more physiologic or morphologic features of a cell. And it's worth noting that this measurement technique is very, very expensive. So to have the fewest number of genes needed to classify cells with the greatest fidelity basically means you have more money left over to measure the genes that you care about for the physiology or the morphology.
Are there any other questions or comments? All right, so last call for questions from the general audience. All right, if not, thanks Sam again for a very nice talk, and I would kindly ask the general audience to leave the Zoom, and just the committee member, please stay here.
Thank you guys all
Thanks
for coming.
everyone for attendance.